Welcome to Ophthalmology Morning Commute, new clinical approaches and sequencing of age-related macular degeneration treatments. Morning Commute is developed by Projects and Knowledge, powered by Kaplan, and is part of a continuing medical education series. This independent CME-CE activity is supported by an educational grant from Genentech, a member of the Rush Group. In this episode, Dr. Arshad Kanani and Dr. Carl Vigillo take a deeper dive into their discussion on treatments of neovascular AMD, including anti-VEGF agents and some of the newer agents, including a bispecific antibody that inhibits both VEGF-A and Ang2. Information about the faculty and disclosures can be found at morningcommutepodcast.com/AMD2. You can use this link to receive your credit and evaluate this program. The URL can also be accessed in the episode notes. Dr. Kanani is Managing Partner and Director of Clinical Research at Sierra Eye Associates and is also a Clinical Associate Professor at the University of Nevada, Reno School of Medicine. Dr. Vigillo is Chief of the Retinal Service at Wills Eye Hospital and a Professor of Ophthalmology at the Sidney Kimmel Medical College, Thomas Jefferson University in Philadelphia. I am your host, Candace Hoffman. Dr. Kanani will begin our discussion.
Welcome back to our continuing series on age-related macular degeneration. I'm Arshad Kanani with Sierra Eye Associates, and it's a pleasure to have my good friend and colleague, Dr. Carl Vigillo from Wills Eye Hospital here. Carl, thanks for joining me.
Pleasure to be here, Arshad. Thank you for having me.
So, Carl, let's talk about the treatment landscape for neovascular age-related macular degeneration and talk about what we did in the past and what we are doing now in terms of transforming how we are treating our patients. So let's start with anti-VEGF therapies. So, you've been in practice for a while and you have seen the transformation that anti-VEGF therapies have done in terms of outcomes for our patients. Before anti-VEGF therapy, we had photodynamic therapy, and before that, we had thermal laser, and patients were losing vision over time. And with anti-VEGF therapy, we were able to maintain vision and to improve vision in many of our patients. So can you provide a little bit high-level overview of treatments, as you call them, first generation, bevacizumab, ranibizumab, aflibercept, and brolucizumab, in terms of the timing and how these treatments were started and how they have transformed in terms of getting better durability and how you're treating your patients in clinic in terms of treat and extend approach versus as needed approach?
Well, as you know, I'm a little older than you, so I've been practicing a while longer, and I've lived the bad old days in practice, the pre-anti-VEGF era. So circa 2006, that's when things really changed for the better. That's when we had the first highly effective anti-VEGF agents, intravitreally administered drugs, hit the scene. Before that, like you said, long ago in the 80s and 90s, early 90s, we had just laser, and it could barely help, maybe 5-8% of our patients. So the vast majority of patients that got wet AMD had disastrous vision loss. Legal blindness was the usual, typical, common outcomes of an eye that turned wet. And that all occurred over a 12 to 18-month time frame. The neovascularization would play out. It would grow, leak, bleed, scar, and then you've got the end-stage scar situation. Now, photodynamic therapy was around the year 2000, and that did help. It was the first time we could treat many of these neovascular complexes in wet AMD, and it minimized vision loss, but patients already had vision loss at presentation. It just reduced the amount of vision loss they would have compared to natural history. So they'd come in at 20/70, 20/80 vision, which was not so good to begin with, and then they'd end up around 20/100, 200, and that was the success story of PDT. But now, 2006, we had bevacizumab and ranibizumab. Bevacizumab is off-label. Ranibizumab was the first FDA-approved anti-VEGF-A biologic. It's an antibody fragment, of course. injected intravitreally. The pivotal studies was injected monthly. And for the first time, we saw mean visual acuity gains in patients that presented with new onset wet AMD. It was historic. It was revolutionary. And we saw 6, 8, 10 letters gained over the course of just a few months of monthly intravitreal injections. And then staying with monthly injections in the clinical trials, we could maintain those gains over 2 years, which is very impressive. So you're talking patients coming in 20, 60, 70, 80 to start with and improving. Many patients were 20, 40 or better, which was remarkable. They could still read, they could still drive, or regain those abilities, we'll say. A few years later, the next FDA-approved drug was aflibercept. And all three of these drugs really are doing all the same thing. They're blocking the VEGF-A isoforms. And that does control wet AMD very well in the vast majority of our patients. Not all of them, but I would say north of 75, 80% of the patients get adequate control of neovascularization with these drugs if administered on a frequent and regular basis. Now, that's the key part, right? Frequent and regular basis, because as we mentioned in the first episode, unfortunately, the real world outcomes outside the clinical trials, patients aren't coming in frequently enough. These drugs are not that durable. In the maintenance phase of therapy, that means after the first 3 or 4 injections, we call the induction phase. In the maintenance phase, all 3 drugs average durability is about 8 weeks, with a range of 4 to 12 weeks. So we can't extend treatment beyond 12. Many of our patients can't go beyond 8. And that means coming into the office frequently to keep on top of the condition to get the best outcomes. So now, a few years later, in fact, just a few years ago, brolucizumab hit the scene, a small single-chain antibody fragment, very potent, seemed to work as well, maybe dry a little bit better and last a little longer, which was key, but we saw some safety issues. Up until then, our anti-VEGFs were super well tolerated, very low adverse event rates with these injections, but we started to see excessive or increased rates of inflammation with brolucizumab. So unfortunately, although it worked very well and lasted definitely longer than the drugs, those first generations, as you put it, first generation drugs that we had been using, it really doesn't get much use because the safety profile is not as good. So in steps the most recently approved, I still call it an anti-VEGF agent because it is. However, there's a second mechanism of action. So that's faricimab. It's a bispecific antibody that blocks VEGF-A like all the other agents, plus it blocks angiopoietin-2. So it's dual mechanism of action. And we think that's a lot of what's behind the additional durability this drug has demonstrated in its pivotal studies. So it's just been in our hands. I know you've used a bunch of it. You can certainly share your experience with it, and I've been using it too. And I think it's living up to expectations of being a more durable anti-VEGF drug with this added mechanism of action to explain that maybe extra drying and or durability.
No, Carl, that's an excellent overview of the treatment landscape. And I think you mentioned some key points there. Number one, that our first generation agents were very effective, but required frequent dosing. And number two, they were very, very safe. So I think any new treatment that comes to the market, I think there's a bar of efficacy, but there's also a bar of safety. And that's why, as you said, brolucizumab obviously met the bar of efficacy and actually better durability, but didn't meet the bar of safety because of risk of vasculitis and retinal artery occlusion. And of course, you mentioned faricimab being a bispecific antibody, blocking VEGF-A and Ang2. And as we know from the preclinical data, that levels of Ang2 are elevated in retinal vascular diseases like diabetic macular edema and neovascular AMD. And what Ang2 does is that it deactivates the Tie2 receptor, which leads to inflammation and vascular leakage. And if we can block Ang2 and stabilize the Tie2 receptors, we hope that we can get greater durability. And as you mentioned, the phase 3 TENAYA and Lucerne studies looked at efficacy, safety, and durability of faricimab with up to Q16-week dosing compared to aflibercept every 8-week per label dosing. And what we saw, the 2-year data that I presented last year, we saw that approximately 80% of patients were on Q12-week dosing or greater at the end of 2 years in the Tenaya and Lucerne study. And almost 60% of patients were at Q16-week dosing. That's every 4-month dosing, which is unprecedented compared to what we had in the past. Now, obviously, we know that for a new agent, we need to have good safety. And in the trials, the safety was comparable. But obviously, the real world data is very impactful and meaningful when you have a lot more patients. And, luckily, in the real world data, we have not seen any new safety signals with faricimab. So it appears that it has better durability and comparable safety. Obviously, efficacy is also important, how quickly we can dry the retina, and if we can help patients who have persistent disease with other agents. And that's why, in the Truckee study, as you mentioned, we have looked at that. And what we saw that for neovascular AMD, we are seeing better anatomic outcomes with dual inhibition with faricimab compared to just VEGF inhibition, whether it was the ranibizumab or bevacizumab or patients who were treated with aflibercept. We're able to dry these patients who had persistent disease, let's say with monthly aflibercept or ranibizumab injections. So I think you and I both know that the disease is very variable and we have to individualize treatment for our patients. But it's nice to have a new agent with dual mechanism of action to kind of help them have better durability. I know, Carl, you've been involved a lot with the port delivery system since the early days of it, and have presented the data and published much of this data over the last several years. Can you tell a little bit about how port delivery system works and just share the top line data that we have seen from the pivotal trials?
Yeah, absolutely. What makes the port delivery system unique is that it's a sustained delivery approach for anti-VEGF therapy. Everything else, of course, is intermittent therapy. And as you said, faricimab sets itself apart from the drugs we have been using, these so-called first-generation agents, because first-generation agents all had very similar efficacy, durability, safety being good, as you indicated. And the dual mechanism of faricimab gives us that added durability and really does set it apart. So that's a great step in the right direction. We're trying to talk about meeting unmet needs, which is drugs with greater durability, duration of action, and therefore less treatment burden. Well, sustained delivery of anti-VEGF means more of a constant level, getting at least a 6-month-like effect. That's kind of what most people would consider as sustained delivery. 6 months or so before you have to redose, or in this case, refill the device. So what this is it's a device filled with a high concentration of ranibizumab. It's 10x concentration, and it slowly releases the device. It's an intraocular reservoir device that we insert surgically into the eye wall that delivers the drug down a concentration gradient into the vitreous cavity. It's slow release, and it provides a good level of ranibizumab, equivalent to about 3 weeks after we do an injection of ranibizumab, approximately. And now we'll control disease adequately in the maintenance phase. That is, after a patient's already been dried up with injections in the office, we then introduce the notion of sustained delivery in the form of port delivery system, FDA approved about 1.5 years ago, started to be used in practice, but it is unfortunately on hold because of some problems related to the septum of the device, which is the septum is how we refill the device in the office. So the device is initially filled and inserted in the OR, and then it's refilled every 6 months or more in the office by putting a special needle through the septum of the device. And it generally works really well. There's no doubt about it. The phase 3 clinical program showed that the device controlled disease in the maintenance phase as good as monthly gold standard ranibizumab injections. Nothing has ever worked better than that. And so this device controlled disease with minimal supplementation, being refilled on a 6-month basis. So it gave us sort of the answer to what we've been asking for, or the questions we've had about trying to get excellent sustained delivery and durability. But it is a surgery, it is a device, and that alone had increased rates of adverse events and issues unique to the device and the surgery. So the safety profile wasn't on par or as good, and that's a trade-off. And some patients are willing to take that trade-off. Now, right now, though, because of the septum issue, that device placement of the device is on hold. So if a patient wanted it right now, they can't get it. We can't implant these devices until the septum dislodgement problem associated with the device is fixed. We hope that happens soon, because I really do think this device is well suited for some of our patients that have a high need for injections, want to decrease the treatment burden, and are willing to trade off some of the increased adverse events associated with having this device and compared to ongoing monthly office-based injections.
That's a great summary, Carl, about the port delivery system. And, based on my experience, I think the patients actually are very receptive in terms of preferring the device. So, I had patients in clinical trials, and once the device went commercial, I actually implanted the fellow eyes of those patients because they were getting injections every 1 to 2 months, and they saw the benefit they got from the device. So I actually have several patients who have bilateral device. I think your point about it being a surgery is important, because not only, a surgical procedure is not going to have the same adverse event profile as an injection. We know that. But I think what I've seen, at least in my practice also, is the learning that over time. So, the surgical learnings about each step, I think have really helped me optimize my surgical outcomes in terms of, having conjunctival retractions or erosions or endophthalmitis and other things. So I think as a field, we are learning how to do the best surgery. But of course, you need to have a device that is, can last a long time. So that's why it's on hold, because of the septum dislodgement issue. And as you said, hopefully we fix it, because there are patients who will look at the risk benefit profile and would like to have the port delivery system instead of having frequent injections. So I think it's a subset of patients who will prefer it, but I think having that as an option is good. I mean, obviously, when we look at sustained delivery, we have gene therapy programs, and we can have a whole episode on gene therapy. So we're not going to dive into the whole landscape, but we have intravitreal gene therapy, we have suprachoroidal gene therapy, we have subretinal gene therapy, and I think being involved with all of these programs, I think what I've learned that it's a different treatment paradigm, and we need to see not only just the efficacy but also the safety. And there have been several learnings about the safety in terms of prophylaxis, as well as what dose to use. So I think those learnings are continuing. Other group of drugs I want to talk for just a short time before we wrap up the episode is tyrosine kinase inhibitors. We have multiple different programs that are looking at it. They're pan-VEGF blockers. They work intracellularly. What are your thoughts just in general, Carl, on tyrosine kinase inhibitors or TKIs?
Yeah, this is in the category. I find them intriguing. I think they're promising. They're all just entering phase 2, so data right now is a little limited. But these are all in the category of sustained delivery, because these are small molecules, tyrosine kinase inhibitors, that if you inject it in the eye, they'd be gone in days. But they're packaged in a way to be slowly released in the eye, either injected intravitreally in the form of a polymer or microspheres or something like that allows the small molecule to be released slowly over 4, 6, 8 months or so, or suprachoroidally as a suspension to again be delivered over 3, 4, 6 months. So we're looking as a durability play. We're looking for an anti-VEGF-like effect, although they're not blocking VEGF directly. Like you said, they're working intracellularly to block the effects of VEGF by blocking the receptor kinase activation. So it's working indirectly, but it's having a VEGF blockade-like effect. And I think it's promising. I think we're seeing some good efficacy some decent durability, 4, 6, 8 months or so in many of the patients, not all of them. It doesn't necessarily control the disease. They're introduced in the maintenance phase, like the port delivery, just like gene therapy, which means we get the disease under control. We know it's responsive and well controllable. And then we introduce something that is of greater durability in a sustained release fashion. So it's like port, it's like gene therapy. And now with these entering phase 2, we'll see, like you said, just like with gene therapy, exactly how safe and how effective they are. They're very different approaches. So we're likely to run into some new adverse events that we might not have encountered before. And we saw that with gene therapy. We saw that with port and the surgery and the device. So as we deviate from injecting biologics, that is antibodies or fusion proteins, we're likely to run into some new issues, which, if they're acceptable, great. And if the products work as well, then we've got another option for our patients.
That's actually a very good summary, Carl, to wrap up the episode. For me, I think over the last 2 decades, we have really made great progress in terms of treating this blinding disease, making an impact for patients around the world, cutting blindness in the world because of these anti-VEGF agents. And now having the first bispecific antibody that blocks VEGF and Ang2, we are able to have greater durability. And hopefully our hope is that we'll be able to maintain vision for many, many of our patients. So with that, I'll close this episode. Thank you, Carl, again, for all the insights you have provided and all your experience. And also want to thank the audience for listening to this episode.
Remember, to receive your credit and evaluate this program, please visit morningcommutepodcast.com/AMD2. You can find all of the episodes in this series and all of our other podcasts on your favorite podcast streaming services, or download our Morning Commute app. Thank you for joining us today.